just a little bit of introduction to myself. I started with Sue Wessler at the University of Georgia in 2008, where we developed a course called the Dynamic Genome. It's an early research experience for undergraduates. In fact, it's now exclusively for freshmen and usually first quarter freshmen who have not had introductory biology at a college level. At UC Riverside, it's 10 weeks. We are on quarter systems in the UCs. The first 5 weeks, we teach basic concepts and techniques. And then the last 4 weeks is a guided research project. And the focus is on genomics. And in a few minutes, I'll tell you a little bit more about our work with transposable elements. In our course, the last project, the research project takes on many different forms. And I'll talk about some of those tomorrow. One that is most common is when we study transposable elements. There are two types of transposable elements, the class 1s, which are the RNA elements, the most abundant type, and the class 2 elements, the DNA elements, which we focus on. They can be as much as 98% of the genome. The iris has almost 2% of its genome as coding for proteins. The rest of it is transposable elements. In humans, it's about 50%, I believe, transposable elements. Usually they are masked and completely forgotten about by genome projects. So there's a lot of potential for students to do some authentic and novel research. The reason that the target was developed initially was so that we could do TE finding in class. Okay, so let me go one more slide. So in class, what we would have to do is they would start with a query, DNA or protein. Then they would do a blast, and they'd sit around for a while, right? You gotta wait for the blast. Then this part is incredibly difficult because you have to copy and paste a whole bunch of blast data and try to put genes back together. Then doing the multiple sequence alignment, again, you're submitting data and waiting. And then you do tree estimation, again, submitting data in a completely different format and waiting. What happens when you're waiting in class and students have a computer in front of them? Facebook. It's no fun. And this could take weeks. Okay, we had 4-hour classes at the University of Georgia. It could take us 2 weeks. Okay, we only now have 3-hour classes at UCR. So Sue went to the grad student at the time, which was one of her students, and said, "Can you automate this?" Well, a year later, we did, and it's called Target. And then, as Eugen was putting together targets, people in the lab kept saying, "Well, I want to use that for my genome. I'm not in class, but I want to use it for my own research." And we went to IPlant and said, "Look, we've got this tool. Can you host it?" And so they have been hosting it since actually the beginning of IPlant. So Target is there. It is an authentic research tool. We use this to annotate the Ambarella genome, another graduate student and myself, and many people are using it. Subway is using authentic software tools. Okay, the only thing that we're really doing is masking a lot of the complexity that students don't need to know about. So the yellow line allows you to, another interface to Target, but it is even simpler. You can either enter a sequence here, or you can select a sequence that's already been made available because of different lessons that are out there. Or you could even ask them to provide them, and they can sometimes add the sequence for you to make it a little simpler for the students. You fill out some information and create your project. On the next screen, you select the genomes that you want to query. Okay, so we start with perhaps a transposase. I'll show you a few examples in a minute. We can query either one genome or multiple genomes at a time. So here I took the ping protein transposase from ping, which is a PIF element, and discovered in Sue Wessler's lab in a collaborating lab in Japan. And we found that this is using the nucleotide query. And you can find here is the original query. Here is the ping that lives in rice, and this is japonica. Here are probably related, or definitely related elements, and these are actually pongs. They're not, they are very, they've been named pong. But then if we take the protein query and do the same genome, look at all the potential homologs we find. So we use our students through the DNA subway barcoding type tools to actually do other mentoring, near-peer mentoring with potential incoming students. And we do this also at the high school level. And we just discussed or set up a project for February the 7th with middle school.